For the final video in the genetics section, I thought it would be helpful to just look at a few genetics problems and work through the solutions step by step. So this first question says, draw it. Pea plants, heterozygous for flower position and stem length, and here their genotypes are given, so flower position is clearly the A gene, and stem length is the T gene, are allowed to self-pollinate, and 400 of the resulting seeds are planted. Draw a Punnett square for this test cross. All right. Well, if I have a pea plant, which is heterozygous for two different traits, in order to make a Punnett square, I have to know what kinds of gametes they can produce, what kind of sperm or egg cells. And since we have two different genes here and two different alleles for each gene, that means that there's 4 different combinations of gametes that they could potentially make. So we have those, we'll say sperm cell, that have the dominant A and the dominant T. We have those sperm cells that have the dominant A and the lowercase t. We have those sperm cells that have the recessive A and the capital T, and we have those sperm cells which will have recessive A and recessive T. Now both of the parent plants are the same, pea plants, which are heterozygous for these two traits. So I can just copy everything I put on the upper side of this square onto the left side of this square. Now, because there are 4 different gamete possibilities, we have a 4x4 Punnett square. A 4x4 Punnett square means that there are 16 possible combinations, but some of them will be equivalent to each other. So, running down the line, I'm just going to put in all of the alleles for one parent first, and then all the alleles for another parent, so that I know that I'm doing it in a kind of organized way. So, capital A, capital T. Okay, so we have all of the genotypes on the 16 square Punnett square for this cross. The next question is, how many offspring would be predicted to have terminal flowers and be dwarf? Terminal flowers and be dwarf. So one thing we need to know is the dwarf variety of this gene is the recessive trait, and the terminal variety of this gene is also a recessive trait. So this question is actually asking how many of these offspring would you predict to have both recessive traits in their phenotype, represented as their phenotype. In order to have both recessive traits, then you need to have 2 sets of recessive alleles for both genes that we're talking about, and only one combination on this chart, that's this one right here, has 2 recessive traits being represented. Now that doesn't answer the question, though. The question is, how many offspring would you predict out of the 400 that are planted? So 1 out of 16 is the fraction of them that we are going to be dealing with. But you need to multiply 1/16 by 400 individuals in order to come up with a prediction. That would equal 400 over 16, which would equal 200 over 8, which would equal 100 over 4, which would equal 25. I didn't happen to have a calculator in front of me, but the answer would be 25 individuals would be expected to have terminal flowers and be dwarf. The next question, what if? List all gametes that could be made by a pea plant heterozygous for seed color, seed shape, and pod shape. How large of a Punnett square would you need to draw to predict the offspring of a self-pollination of this tri-hybrid? Ooh, okay, so this is a somewhat tedious question, but it's absolutely answerable. The first thing we have to do, they haven't given us letters to assign for these genes, and it actually doesn't matter what trait is dominant and what trait is recessive, because that's not the question that we're answering here. But seed color, we're going to give a capital Y for yellow, a lowercase y for green. Seed shape, I'm going to give a capital R for round and a lowercase r for wrinkled. And pod shape, I'm going to give a capital I for inflated and a lowercase I for constricted. Those are all of the possibilities. Now, knowing which of those traits exist and which ones are dominant, which ones are recessive, are not actually relevant to answering the question in front of us. The question in front of us is really just it's a math problem when you get down to the core of it. But this, we have a pea plant that's heterozygous for all of these traits. This is its genotype right here. Heterozygous for seed color, heterozygous for seed shape, and heterozygous for pod shape. The question says, list all gametes that could be made by a pea plant of this type. Okay, so they actually want us to list them all out. If you want to know how many possibilities there are total, though, we're going to make some gametes from this plant. Each gamete, sperm or egg cell, is going to have just one copy of each of the allele pairs in the parent. So they're going to have 3 alleles at the end, one of each type, for which there is 2 possibilities for the first one, capital Y and lowercase y. There are 2 possibilities for the second allele, and there are 2 possibilities for that third allele. That is equal to 2 cubed, which is equal to 8 different possibilities. Now, right away, how large of a Punnett, the next part of this question, how large of a Punnett square would you need to draw to predict the offspring of a self-pollination of this trihybrid? Self-pollination means that essentially we are breeding this plant with itself. So both of them, both sides of the Punnett square will have the same number of possibilities, which is 8, which means we would need, I can already answer this one, it would be an 8 by 8 Punnett square, which is a 64 square Punnett square, which is a chessboard, as it turns out. So you would need an entire chessboard in order to lay out all of the possibilities here. But the first part of this question is still unanswered. It says list all gametes that could be made by this pea plant. So we know there are 8 possibilities. 1, 2, 3, 4, 5, 6, 7, 8. So I'm just going to run through all of the possibilities. In order to do this, you're going to want to organize your thoughts. You're going to want to do it in a precise way. So I will do everything that has a capital Y at the beginning first. So capital Y, capital R, capital I. That's my first possibility. Capital Y, capital R, lowercase I. Possibility 2. Capital Y, lowercase r, capital I, capital Y, lowercase r, lowercase I. That's all the possibilities that exist with just a capital Y at the beginning. Everything else is going to have a lowercase y at the beginning. So we have lowercase y, capital R, capital I. we have lowercase y, capital R, lowercase I. We have, that exhausts all of our capital R possibilities. So capital I, lowercase I. Those are all 8 possible sperm or egg cells, or in this case both, because it's a self-pollination, that could be made using this particular plant. Next question. Incomplete dominance and epistasis are both terms that define genetic relationships. What is the most basic distinction between these terms? Incomplete dominance is a situation where if you have the homozygous dominant version of the genes, you get trait number 1. If you have the homozygous recessive version of these genes, you get trait number 2. But if you have the heterozygous version of this trait, you get something that's halfway in between. You get trait 1.5, so to speak. So if trait number 1 is having black fur, and trait number 2 is having white fur, if you have a heterozygote for this gene, they are going to have gray fur. Epistasis is the situation where the occurrence of a gene at one location changes how a gene at another location is represented as a phenotype. So the example that I ran through was with dog fur. So you could have a gene that says dominant is, this is the black fur allele. Recessive is, I'll call it the red fur allele, because that's how dog breeders discuss it. I think I may have said brown fur or chocolate fur in one of the previous videos. Okay, and those are your possibilities. Now, this doesn't have incomplete dominance, so these two genotypes both get you black fur. Only this genotype gets you red fur. The epistasis comes in when we add in a second gene. So we have the dominant E allele, which says make fur pigment. And the recessive E allele, which says no pigment. This set of alleles affects these sets of alleles. They determine whether or not you can even detect what the phenotype, what the genotype for that set of alleles is in the animal. So if I were to have, right, we have a dog here who has two dominant alleles for black fur, capital B, but they also have two recessive alleles for pigment production, you will not get a black-furred dog. As a result, you will get a yellow lab. Because that is what a Labrador Retriever looks like when they do not produce pigment in their fur cells. So, back to the question. What is the most basic distinction between these terms? I would say the most fundamental distinction is that incomplete dominance deals with the relationship of a single family of alleles, a single type of gene, a single locus of genes. Whereas epistasis deals with multiple loci of genes, two different families of alleles, two different genes affecting each other. If a man with type AB blood marries a woman with type O, what blood types would you expect in their children, and what fraction would you expect of each type? Okay, so type AB blood, this is a codominant situation. You have three alleles, capital IA, capital IB, and lowercase I. The woman with type O blood, that is the phenotype you get when you have two recessive alleles. The man with AB type blood has one of each kind of dominant allele. So we can do an easy Punnett square for this cross here. This is the woman's alleles. This is the husband's alleles. And the mother has no choice but to give every single offspring one of her recessive alleles. 50% of the man's offspring will get a dominant A, capital IA allele. 50% will get a capital IB allele. What fraction would you expect of each type? So we will get this person has A-type blood because they have the A allele and a recessive. This person has the same phenotype. This person has B-type blood. Same here. So you would get one A-type to one B-type. It's a 1-to-1 phenotypic ratio. What fraction would you expect to see of each type? 1/2. 1/2 of the offspring would have each type. You would have 0 offspring with AB type blood. You would have 0 offspring with type O blood. You would only see A type blood and B type blood from this cross.